Welcome to DermWire News, powered by PracticalDermatology.com. I'm Stephanie Talea, Senior Managing Editor. Cutera is launching the new TrueSculpt Flex muscle sculpting platform in the United States, and commercial shipments are slated to begin immediately. TrueSculpt Flex is FDA cleared for the indications of the improvement of abdominal tone, strengthening of the abdominal muscles, and the development of firmer abdomen. It is also cleared for the strengthening, toning, and firming of buttocks and thighs. We are very excited to introduce TrueSculpt Flex and proud to be the first aesthetics laser company to offer a muscle toning system that treats up to 8 muscle groups simultaneously. Covering the largest treatment area in the body sculpting industry, says Jason Ritchie, COO and interim CEO of Cutera, in a news release. The TrueSculpt Flex technology is further differentiated from other devices through our proprietary multi-directional stimulation, or MDS technology, that delivers 3 treatment options in one session by replicating intensified crunch, squat, and twisting actions. Further, he continues, with the launch of TrueSculpt Flex, we are expanding our body sculpting product family as the TrueSculpt Flex joins our TrueSculpt ID fat reduction technology launched in 2018. During the clinical study for TrueSculpt Flex, our practice was very pleased with how the technology performed and delivered real personalized muscle toning results for patients, says Washington, D.C.-based facial plastic surgeon Michael Somanek. He was an investigator for the TrueSculpt Flex clinical trials. This technology opens up the possibilities for any aesthetics provider to offer top-of-the-line muscle toning services. With TrueSculpt Flex, 8 muscle groups are treated simultaneously, covering the largest treatment area in the body sculpting industry. 3 treatment modes in the same session simulate different workouts by replicating intensified twisting, squat, and crunch actions, Cutera states. Prep mode creates a twisting motion to warm up, stretch the muscles, and slowly build a tolerance to muscle contractions. Tone mode contracts the muscles, holds it to the point of exhaustion, and then relaxes it to increase strength and enhance endurance. Sculpt mode, fast, deep, sequential contractions of the muscles to develop new muscle fibers and increase basal metabolic rate for building muscle mass and reducing the circumference. The proprietary MDS technology involves direct delivery of electrical current to stimulate muscle contractions without energy wastage and treat specific muscle groups using 3 treatment mode options. The device offers customizable handpiece configuration and placement locations to target multiple specific small and large muscle groups with no downtime. These low levels of energy achieve deep, full muscle contractions at high intensity. Columbia researchers have created a way to use 3D printing to create hair farms in a dish, which could open up hair restoration surgery to more people, including women, and improve the way pharmaceutical companies search for new hair growth drugs. The findings, which appear in Nature Communications, represent the first time that human hair follicles have been entirely generated in a dish, without the need for implantation into skin. For years, it's been possible to grow mouse or rat hairs in the lab by culturing cells taken from the base of existing follicles, the researchers report. Cells from rats and mice grow beautiful hair, says study author Angela M. Cristiano, the Richard and Mildred Rodeback Professor of Dermatology and Professor of Genetics and Development at Columbia University Medical Center in New York City. But for reasons we don't totally understand, human cells are resistant. To break the resistance of human hair cells, Cristiano has been trying to create conditions that mimic the 3D environment human hair cells normally inhabit. The lab first tried creating the little spheres of cells inside hanging drops of liquid, but when the spheres were implanted in mice, the results were unpredictable. The cells from some people created new hair, while others didn't. In the new study, Cristiano's team exploited the unique capability of 3D printers to create a more natural microenvironment for hair follicle growth. The researchers used 3D printing to create plastic molds with long, thin extensions only 0.5 millimeter wide. After human skin was engineered to grow around the mold, hair follicle cells from human volunteers were placed into deep wells and topped by cells that produce keratin. The cells were fed a cocktail of growth factors spiked with ingredients, including JAK inhibitors, that the lab has found stimulates hair growth. After 3 weeks, human hair follicles appeared and started creating hair. Though the method needs to be optimized, engineered human hair follicles created in this way could generate an unlimited source of new hair follicles for patients undergoing robotic hair restoration surgery. What we've shown is that we can basically create a hair farm, a grid of hairs that are patterned correctly and engineered so they can be transplanted back into that same patient's scalp, Cristiano says. That expands the availability of hair restoration to all patients, including the 30 million women in the U.S. who experience hair thinning and young men whose hairlines are still receding. Hair restoration surgery would no longer be limited by the number of donor hairs. The engineered follicles also could be used by the pharmaceutical industry to screen for new hair growth drugs. Currently, high-throughput screening for new hair drugs has been hampered by our inability to grow human hair follicles in a lab dish. No drugs have been found by screening. The only two approved for the treatment pattern of hair loss, finasteride and minoxidil, were initially investigated as treatments for other conditions. The team hopes that cultured hair farms will open the ability to perform high-throughput drug screens to identify new pathways that influence hair growth. Thank you for listening to DermWire News, powered by PracticalDermatology.com.